Okay, the three stages of translation then are, since we're now moving past all the materials involved, like messenger RNA, transfer RNA, and ribosomes, we know that they are the major components of the process, but now we need to get into what are the stages of translation. And so we have three stages: initiation, elongation, and termination, just like we had in transcription. However, to make all of these processes happen, in initiation you needed one, but we talked about, in transcription we needed transcription factors, but in this process every stage requires factors, protein factors, that help in the process. And so to get this process to start, initiation begins when messenger RNA and a transfer RNA that has the correct first amino acid, as well as then the two ribosomal subunits interact with each other. So the large subunit and the small subunit are floating in the cytoplasm. The messenger RNA is released from the nucleus, and the tRNAs are floating around in the cytoplasm. So the first thing that happens is that small ribosomal subunit is going to find the messenger RNA, and it's going to bind to it, as well as with a tRNA that is an initiator that begins the process. So that initiator tRNA, the messenger RNA, and this small ribosomal subunit bind together. That small ribosomal subunit then moves along our messenger RNA until it runs into the start codon, AUG, which is not the same thing as the special initiator tRNA. The start codon is on the messenger RNA, and when the small subunit interacts with it, then initiation factors will come in and bring with them the large subunit, the large ribosomal subunit, that then begins and completes that translation initiation complex. So the small subunit binds to the messenger RNA along with this special initiator tRNA molecule. The small subunit then is going to move along the messenger RNA until it hits that start codon. When it hits the start codon, initiation factors will bring in the large ribosomal subunit, completing that translation initiation complex. So here we see how we start, right? The start codon is downstream of where we actually bind, right, with my initiator tRNA. When it hits the start codon, then the large ribosomal subunit comes in and begins the process of translation. So elongation of the chain, then during elongation the amino acids get added one at a time to the existing amino acid chain. Elongation factors are the proteins that are involved in this, and to make elongation occur there are three steps in the sequence. Codon recognition, so you have to know what codon you're engaging. Peptide bond formation, because when the next tRNA with the correct amino acid comes in, you have to form a bond between the existing chain and the new amino acid. And then translocation, which is the movement of the ribosome down the messenger RNA. So here we have our tRNA in the P site with the amino acid chain, and we are bringing in the next tRNA carrying the next amino acid is engaged in the A site. So that would be codon recognition in that first step. Then in the second part, we see the peptide bond forming there between the existing polypeptide chain and that amino acid. Then we have the chain complete, and that whole polypeptide chain is on the tRNA in the A site temporarily until translocation happens. Translocation allows the A site to be opened by kicking out the empty tRNA out of the E site. And this process continues. Codon recognition, polypeptide bond formation, and translocation keeps going until you hit a stop codon. And when that stop codon is reached, when it hits the A site of the ribosome, the A site receives a release factor. So another type of protein, right, translation factor, called a release factor. This release factor binds in the A site, and that binding causes a water molecule to be added instead of the amino acid. That reaction basically breaks apart the translation assembly. And the ribosomes let go, the tRNAs let go, the polypeptide chain is cleaved from the tRNA that was holding it, and the process comes to an end. So there's my stop codon, and when that stop codon is in the A site, a release factor binds to that, the polypeptide is freed, and the process, all the parts come undone. A polyribosome, one way that we can get a lot of products made, how do we get, an adrenaline rush? How does that occur? How do all those chemicals get made at one time? Well, it happens because we can actually, make a whole lot of messenger RNA at one time, but then you can also translate the same messenger RNA sequence repetitively with lots of ribosomes. So as soon as one ribosome clamps down and starts moving along translating, another ribosome will also engage that same messenger RNA. We call this process, or this structure, this occurrence, a polyribosome. And that is simply when lots of ribosomes translate a single messenger RNA, and that allows us to get a wave of polypeptides made very quickly for a response to some kind of stimulus. And so here's an image of our polyribosomes with all the ribosomes on the messenger RNA, making lots of copies of our completed polypeptide.